Hi everyone, congratulations for completing week number 1, which was chapter 17, Blood. I'm going to review that chapter with you a little bit, but first I want to make sure you understand that the graded activities were from assignments, the PhysioX blood analysis activities 4 and 5 from on Mastering A and P. That was a graded activity. And then your Chapter 17 quiz from Mastering A and P was also a graded activity. Now, we are going to go over some of the information from the Chapter 17 slides, but first we're going to week 2. This week you have 2 chapters. The graded activities from Chapter 18 are the PhysioX blood analysis from Mastering A and P. This is part 2, so it's not activities 4 and 5. I think it's the first 3 activities, but it's just different activities from that same lab. And then the Chapter 18 quiz, also in Mastering A and P. Notice that this is a link here, so you can just click straight and get to Mastering A and P from this link. The Chapter 19 assignments are very similar. All of this is due on December the 13th, Chapter 18 and 19. Chapter 19 also has a PhysioX assignment and a quiz. So this is due by December the 13th. Both activities are set to where you can do them up to 5 times. You have 5 attempts on these activities. So if you don't make a grade that you are happy with, then you can go back and redo and try to increase your grade. And this also helps you with studying for the midterm exam. Now I'm going to go to the Chapter 17 slides real quick, and I was able to look at your Chapter 17 quiz and from there figure out some of the questions. I want to go to let's go to the Hematocrit first. Here we go. I wanted to blow it up. So basically what a hematocrit is when they draw blood from the area where the bend of your elbow, which is what we also call the antecubital region. Remember from Bio 168? When they take that blood sample, they're going to put it in a test tube. And sorry, by the way, this vein is the median cubital vein. That's the vein that they typically draw blood from, is the median cubital vein. And then what they're going to do is they're going to centrifuge the blood sample. It's going to spin that whole blood really, really fast so that the red blood cells, or erythrocytes, will fall to the bottom because they're heavier. And then you'll have the plasma at the very top layer because the plasma is the lightest. It's the fluid component of the blood. And then the buffy coat, it's called the buffy coat, is white, and it's in the middle between the plasma and the red blood cells. And it contains white blood cells and platelets. So leukocyte is another name for white blood cell. So one of the questions that you had on your Chapter 17 quiz asked, what if the buffy coat was extremely thick? And that would mean that you have a large amounts of leukocytes and or platelets, and a large amount of white blood cells would indicate severe infection. So I wanted to make sure I went through that one with you and the hematocrit with you. And now we've talked about erythrocytes, or red blood cells. Now let's go back and look at the part of the red blood cell that carries oxygen, because I'm sure you know that your red blood cells, one of the functions is they carry oxygen throughout your bloodstream. So this is a red blood cell, a mature erythrocyte. Looks kind of like a donut that hasn't, the hole hasn't pushed all the way through. But this is a picture of the hemoglobin. Red blood cells contain many, many hemoglobin molecules. This is a protein composed of 4 separate globular proteins. 2 beta globin chains they're represented by the dark brown proteins here, And then 2 alpha globin chains are represented by the light brown proteins. So you've really got 4 globular proteins that are connected together. That means this is the quaternary structure of hemoglobin. And each of your globular proteins contains a heme molecule. And that heme molecule is like this, it's this red disc here. The heme molecule also contains an iron atom, and the iron atom in the center is actually what attaches to oxygen. So due to the fact there are 4 heme molecules and 4 iron atoms, then one hemoglobin protein can actually carry 4 oxygen molecules. Let's see, I'm looking in my notebook at some of the questions that you had. We went over the components of the hemoglobin. What happens to the red blood cell? Because red blood cells or erythrocytes, basically this is how they start. They start as a stem cell called a hemocytoblast. Hemo means blood, cyto means cell, and blast means any cells that are going to form into new cells. Remember, osteoblast produce new bone tissue. So that's going to form into a proerythroblast and go through several stages until it becomes a reticulocyte. And at this point, around about the time it forms into a reticulocyte, it loses the nucleus and the mitochondria, and then it becomes an erythrocyte, and that erythrocyte has a shorter lifespan. It's only a couple of months, a few months that the erythrocyte actually can live before it's recycled in the spleen. So let's talk about the life cycle. Here we go. The life cycle of a red blood cell. If you look at the stages, the red blood cell, red blood cells actually come from the red bone marrow. So that's found in the spongy bone. And number 5 says aged and damaged red blood cells are engulfed by macrophages of the spleen, liver, and bone marrow. So that's where red blood cells are recycled. In the spleen is one of the primary areas. Okay, let's see. Components of the hemoglobin, the red blood cell graveyard. We talked about the Buffy coat. Erythropoiesis is the generation of blood cells. And, okay, so this slide, I don't think I need to blow it up that much, is just telling you that EPO is a hormone produced by kidneys. Its long name is erythropoietin, but it is produced by the kidneys, and whenever the kidneys produce EPO, then erythropoiesis occurs, and that means more red blood cells are made. And this normally happens because the cells of the kidney recognize a condition called hypoxia, or low oxygen. Here we go. Okay, it says the stimulus is hypoxia or low oxygen in those kidney cells, and that will cause the kidney cells to release the hormone EPO or erythropoietin, and then that will lead to the red bone marrow that's found in the ends of long bones to produce more red blood cells. So that's the relationship of EPO to erythropoiesis, and that EPO again is produced by the kidney. When a person is dehydrated, so what does this do? If a person's dehydrated, I think it's best to go back up to this hematocrit, and remember, if you're dehydrated, this plasma level is going to be lower than what it is. It's going to be less than what it is showing here. So you're going to end up with lower plasma levels. You're going to end up with higher blood viscosity because this whole blood comes from the mixture of the plasma and all of the blood cells, and it's going to be thicker with less of the liquid part. So thicker means higher viscosity. You're also going to have a higher hematocrit. You're going to have a higher red blood cell level compared, the hematocrit is named for the percent of red blood cells. And so you're going to have more of a percent of red blood cells than you are to plasma. So maybe instead of 45%, your percentage of erythrocytes goes up to 55 and your plasma goes down to 45. You know, that's going to indicate dehydration. But what it will not do is lower your immunity, because that really, the dehydration really doesn't affect this buffy coat here, or the white blood cells. Okay, if we go back to blood cell formation, let's find one that shows, there should be a chart that shows the formation of all of the formed elements of the blood. We looked at one that showed just the red blood cells, but let's look at one that shows all the formed elements of the blood, including white blood cells and platelets. Well, since we got to this first, we'll talk about these, and then we'll do the formed elements. All right. So some of you missed. You were supposed to identify the white blood cells. Leukocyte is the name for white blood cells. There are 5 main types of leukocytes. The neutrophils, and you can identify them a lot of times by their nucleus. This nucleus is not the typical round nucleus that we're used to. It's a multi-lobed nucleus. It has many lobes, so it's a multi-lobed nucleus. These are neutrophils. Eosinophils are going to have all these red granules in them, as well as a bilobed nucleus. And the neutrophils are like the first responders to infection. The eosinophils are really, just to give you an overview, they're you're going to find a lot of eosinophils if you have a parasitic infection. And basophils release histamine, so they give us our allergy symptoms. And the basophils are going to they're going to have granules in them. They do have a bilobed nucleus, but it's not, you can't always see it as well. Lymphocytes are small. They're about the same size as the red blood cell, and you see the dark nucleus almost takes up the entire space of the lymphocyte. Lymphocytes are the ones that we also call them T cells and B cells. These are the cells of our immune system. Then monocytes become macrophages, and they're going to have a large kidney-shaped nucleus. They're going to be the largest of the white blood cells. Now let's go to blood cell formation. If I can try to do this before, here we go. This is what I was looking for. Okay, so this is just a table that shows us the formation of all of the formed elements. So if you look here, you can see that the hemocytoblast is the stem cell for all blood cells, and it will it will either form into a myeloid stem cell or a lymphoid stem cell. Only the lymphoid stem cell can produce lymphocytes, B and T lymphocytes. The myeloid stem cell can produce all the way down here, it can produce the eosinophils, basophils, neutrophils, and the monocytes, as well as the red blood cells and the platelets. So the question says which stem cells give rise to leukocytes? Well, it is going to be the myeloid stem cells that give rise to all of the leukocytes besides the lymphocytes, and that will be the lymphoid stem cells. Okay,